Hello, everybody. I am really pleased to host Dr. Giorgio Gilestro to our International Sleep Charity talk. Dr. Gilestro is from Imperial College London and is currently a senior lecturer. He's also one of my mentors, and I've been really pleased to work under his guidance for a few years, actually. So without further ado, I'll let Dr. Giorgio begin his talk. And thank you again for joining. And thanks again, Dr. Gilestro, for agreeing to speak to us.
Thank you, Gizu. Thank you, everyone, for the invitation, for being here. So what I want to do now in the next 45 minutes or so is to give a kind of an overview of what we've been doing in the past few years, and particularly doing this with the goal of showing that even from fruit flies, from humble fruit flies, we can learn about sleep to an extent that is actually very fundamental. Normally we used to think about fruit flies in terms of their genetics, which is clearly powerful and well known. And then normally, from the genetics, we try to extrapolate what might happen to humans or other animals. But there's much more we can do when we look at fruit flies in the terms of evolution. And if we do take an evolutionary approach to our quest, to our questions, everything becomes more powerful, even without going into genes. And I'll show you why, hopefully. So why evolution? Evolution is really a critical aspect of anyone who will want to study sleep. It's really important to think of sleep in terms of evolutionary conservation. And it's natural to do so because it's frankly puzzling the fact that sleep is so well conserved among animal species. It's been said, that's what you normally read in reviews, that sleep has been found in all the animals that we've studied so far. And that's true. However, it's true with one caveat, that the sleep amount that we recognize in different animals can vary. It can vary dramatically between extremes. Here in these slides, you have two examples of these extremes, a bat on the left and an elephant on the right. Now, elephants are among the animals that are shown to be sleeping the least. They are not the only ones. There are many like elephants, like giraffes and even horses, animals that sleep as little as 2 hours, 1.5 hours a day. Bats, on the other hand, are animals which are believed to be sleeping almost the longest of any known conditions. Obviously, studying bat is always complicated, and the data we have about bat is limited. But the ones we have talk about sleeping pattern that can reach up to 19 or even 20 hours a day. So these are two extremes, two mammals in a way, one sleeping up to 20 hours a day and the other sleeping 2 hours a day. So this variability of sleep amount, if you think about it, is very puzzling. And it is a problem for anyone like me or any sleep scientist who will try to put forward an hypothesis of sleep function. Because any hypothesis of sleep function that we can think of must come to terms with this variability. Any hypothesis of sleep function must explain why an animal needs 20 hours of sleep and another animal needs 2 hours of sleep. If we postulate that sleep does something which is biologically fundamental, then you have to explain why this something can be done in 2 hours with an animal, but takes 20 hours in another. And even without going to these extremes, it's clear that there are major differences in sleep amount. And these major differences, which I reported here, taken from this review, for instance, highlight variability, but do not necessarily highlight conservation. So it's difficult just by looking at the phylogenetic tree to predict whether an animal will be a long sleeper or a short sleeper. So it doesn't seem to be clearly obvious whether an animal will sleep more on the 2-hour side or more on the 20-hour side. There's another aspect of this variability, which is very interesting, and that is that even within the same species, you do see variability in this case, for instance, with migratory birds throughout time. So migratory birds are a fantastic example for this. They're a beautiful species to study. What they do is, as you know, they migrate at one time during the year. Lots of studies have been done on birds that migrate from South America to Northern America, to Alaska and Canada. And these are birds that migrate during the mating season. So they have to start a very long trip lasting weeks. During this trip, they are reported to be reducing the sleep amount by almost 95%. So using EEG and miniaturized computers that this bird can carry on top of the head, scientists can, like Ms. Rattenberg in Germany, could detect that sleep amount in this animal is reduced dramatically by 95%. So that's a staggering decrease if you think about it. That goes on spontaneously for weeks. And when these birds reach their target, in this case Alaska, because they fly for mating seasons, they obviously have to compete for females and for partners. And so what they do is they try to stay awake again for as long as they can, just to increase the chances of competition. It's also been reported that after mating, they will start flying again towards Russia, often again in a pattern of lack of sleep that in this case then can last for months. So how comes that these animals can, with a snap of a finger, almost completely remove their basic instinct for sleep, and they can suppress almost completely their need for sleep for weeks or months every year without suffering any apparent consequences? So that's, again, another example of staggering variability, which is very difficult to explain in terms of sleep function. The same caveat applies. Any hypothesis of sleep function must come to terms with this phenomenon. If we do postulate the sleep as a function X, then we have to explain why these animals can go without function X or without associating function X to sleep for so long. So obviously, the explanations that you can come with when facing puzzles like these ones are let's say, of two kinds. If you really want to reduce to basic principle, you can only think of two possible explanations. Either nature has found a way to sleep more efficiently. So nature has found a way to condense into 2 hours what another animal needs 20 hours for. Or maybe nature has found a way to condense into few minutes what a bird will normally do in several hours. So either that, or sleep serves different function in different species at different time, and this different function can be modulated and suppressed in different manner. Now, explanation number one is still problematic, because then you might argue, if nature has found a way to sleep to sleep more efficiently, if nature really has found a way to condense our sleep need from several hours to 2 hours or few minutes, then why don't we always do it? Why don't we just sleep a few minutes a day, all of us, all of the animals in the animal kingdom? If that efficiency exists, why don't we take full advantage of it? Why only some animals, in fact, do take full advantage of it? So the other explanation on the other end, which is the one I want to push forward, will offer many more answers than problems. And this is what I've been doing in my laboratory for the past several years. And we came up at the end with a model of sleep function which take all these puzzles into account. And we call this tripartite model of sleep function. The basic principle is that we should not think of sleep as a monolithic phenomenon. It's not just sleep, one function, if you want. Sleep is divided, in fact, into different epiphenomena, and each one of them linked to a different function. And when I'm talking about different function, I'm talking by first principle in general terms, not specifically about cellular or biological functions. So I here have a breakdown of what I mean by this tripartite, by these three components. The idea is that part of the sleep we have every night is elemental or basic. So that is the sleep that is associated to a major yet mysterious biological function. Think of this as something as really as fundamental as you can go when it comes to cell biology. DNA repair or whatever, cell membrane repair, or mitochondria clearance, or cleaning residues from tissue. So whatever it is that as a cell biological basis, this is probably something that will be a good guess for an elemental function. On top of an elemental function, you then have a useful function. So useful means that it's something that is important, but is not vital, as elemental will be. So examples of this can be, for instance, consolidating memory or reinforcing the immune system. Those are all things which are really useful, really important, but not vital from the cell biological point of view. And finally, you have a component of sleep, which I'm going to propose is actually the largest majority of the sleep we get, which is completely accessory. And the only function of this sleep is actually to stay out of trouble. So stay put, save energy, don't venture in places which you should not venture at night. And basically, that will be the default state for an animal in order to remove and reduce the chances of danger or peril, or even just to reduce energy consumption by going around. So if you put these three things together, then you can suddenly explain all these differences we saw so far. You can explain the differences between a bat and an elephant, but you can also explain the differences within migratory birds. And the idea is that some animals have a largest amount of accessory sleep, and some other animals have managed to erode on that. So why would elephant or cavefish sleep very little, 1 or 2 hours a day? Well, for elephant, one explanation could be that those are huge herbivores, so animals that have to sustain a high intake of calories, and so they cannot afford to stay asleep for most part of the day. They have to eat. On the other hand, bats are small carnivores often, and they can get a good energy boost by a good meal. They can satisfy their social needs and then go back to sleep and save energy. And so by modulating basically their need for accessory sleep, you can explain this extreme. The other advantage of this system is that now we can start thinking about functions of sleep without fighting, without each one of them fighting for the function of sleep, because we can now think of the whole aspect and break it down and assess different functions which were difficult to explain otherwise from the evolutionary perspective. Like potentiating learning and memory. It's easy to explain why, or to figure out why human might have a big advantage from consolidating memory, but it's more difficult to do so. To explain why the same advantage should be equally important for a C. elegans or a Drosophila. So having said this, I'm going to give examples of how we then break down this problem. And again, we can, let's say, divide the sleep analysis within several aspects, and the ones that are considering here and talking about here, which are absolutely not the only ones, but I believe are probably the most important ones, are sleep amount. So how much do we sleep? So what is the difference between, again, the sleep of a bat and the sleep of an elephant? Sleep quality, which often is made to be recognized by reversibility of sleep, right? We often talk about sleep depth in terms of light sleep or deep sleep, and that correlates very much with our ability to wake up to external stimuli, for instance. And finally, homeostasis, which is the process which regulates everything that concern sleep. So from sleep amount, but in a certain sense also sleep quality. So let's start with the easiest aspect, which is sleep amount. And I'm going to show some data that we collected in Drosophila over the year, which were quite surprising. In fact, are the data that really started our path towards this tripartite model of sleep function. So we use flies, and I hopefully expect most of you to be familiar with the animal model. I just want to remind you that the reason why we use flies is because we believe they they do sleep. They show all this typical characteristic of sleep. They satisfy the textbook definition of what sleep is. So sleep in flies is a period of quiescence which is associated to a specific posture. It matches an increase in arousal threshold, meaning if the fly is awake, even just approaching the fly will elicit some response. But if they are asleep, you need a stronger stimulus to wake them up, exactly as it happens with us. This reversibility to wakefulness is quick, which distinguishes sleep from coma. And finally, there is an important homeostatic control. Homeostatic control basically means that, like in humans, also flies, if they stay up all night because they go to a party or because they have to study, then they'll be more tired the day after. On the other hand, if they have an extra long nap in the afternoon, they will have a bit more of a hard time falling asleep during the evening. And finally, all this shows interaction with the circadian clock. You also get some bonus aspects like conserved signaling pathway, in general conserved pharmacology, meaning that if you give drugs to melanogaster that are known to elicit or inhibit sleep in humans, you will see the same thing in flies. So how do we study sleep practically? When I started my sleep research, really the only system that was used routinely was a system that was inherited by the circadian science. And it's a system that I show here, which is often referred as the infrared beam split system. The idea is simple. You have a single fly in a glass tube, and the fly can stay in this glass tube for days or maybe even weeks. As long as the food is accessible, the fly is relatively happy here. But all she can do is really moving back and forth within the tube, which they do happily every day. As they do move back and forth, what they will do is they will break this infrared beam, which obviously cannot see, but the computer can detect this movement, can detect this breaking of the beam. And so the computer can count how many times every minute the fly will have crossed the middle of the tube. And this system has been really instrumental to study circadian rhythms. It's been good enough to win 3 Nobel Prizes, which we know, we won, the field won in 2017. So it's pretty good system to study circadian rhythms, but it's not good enough to study sleep. And the reason you can think of it intuitively is that if the fly stays awake away from the beam, then any moment that basically the fly is spending away from the beam can be, in principle, misinterpreted as an excess of sleep. So if the fly is by the food, having a large consistent meal, or if the fly is by the cotton, thinking about life existence or whatever, then all this will be interpreted as sleep, while it's not. So to improve the system, to improve the detection, we invested quite a bit of time in creating a new device, which is based on the combination of mostly open source, off-the-shelf component. And the 2 really brains of the system are a Raspberry Pi, which is a microcomputer developed in Cambridge by the Raspberry Pi Foundation. It is a real computer. It's a computer you can connect your monitor and internet and keyboard to and mouse, and you can do Facebook and Twitter and all the things you do with a computer, but it's extremely cheap. It only costs about 20-30 pounds. And the other aspects, the arm of the system, where the Raspberry Pi is the brain, is an Arduino controller. Arduino controller is a microprocessor which can be easily programmed for purposes of creating robots mostly. And so if you go on YouTube and look for an Arduino project, you will find a long list of DIY robotic projects, which are really fun and can make great weekend project for everyone. So when we put these two things together, then we end up with this machine, which we call a ethoscope. The ethoscope has a Raspberry Pi here on top, connected to a camera, and the camera will look down where the flies are. The flies are here in these tubes. And so the basic principle is that now we are not relying on crossing an infrared beam anymore, but what we're doing is we're looking at the flies at every single moment, and we therefore know what they're doing, and we know where they are, we know whether they are interacting, we know whether they're eating, and so on. And so the resolution that we get of their activity is way more reliable. So we published this in 2018, I think, and as everything else we do in the lab, we really put a lot of effort in making sure that everything we do is accessible, so everything is open source. The plans for building endoscopes are available from the very early beginning. Here as examples of how you could build an endoscope if you wanted to. Normally, we use 3D printed devices like the one here, depicted here in the cartoon. But the paper also has instruction to build ethoscopes out of paper, cardboard. We just fold and glue things together. And there's even an instruction to build one out of Lego. In fact, this is the supplementary material of the paper that you can download from the PLOS Biology website. And it will guide you through how to build a fully functioning Lego ethoscope. Unfortunately, we did have to remove the Lego logo for copyright reasons, but we did manage to sneak in the Lego minifigure in the bottom. So when we look at sleep in fruit flies using our system, what do we see? Here is a comparison using many hundreds of flies, and comparing the sleep amount and pattern between ethoscopes and infrared beam split. So this is in a way, is a system that looks at the very same animal, and the same animal is then the sleep in the same animal is then assessed by both systems. And what you see is that the sleep pattern in male and female is relatively similar, independently of what system you use. So you do see a increase of sleep during the night. So flies do tend to sleep mostly during the night, which makes a lot of sense for animal that flies, insects in general. Males specifically have also a nap, so a daily sleep pattern, which we call a siesta, which is not as present or as staggering in females. So in terms of the pattern is similar, but in terms of the amount, as you can see, there is a large difference. And when you quantify this large difference, you will see that with the infrared beam split, you would think that male flies sleep for more than 80% of the time, while in fact they sleep only for 50% of the time. And on the other hand, female flies, you will think they would sleep for 60 or more % of the time, while in fact they sleep for less than 25% of the time. So you can basically easily overestimate your sleep amount by 2 or almost 3 times if you don't use the proper tool. So having the confidence that now we're looking at sleep with a greater resolution and with a system that we can trust more, we then embarked into an analysis of sleep in just wild-type laboratory flies, so Cantonese flies, in large numbers. And so here you see the analysis of sleep amount in about 500 male flies and almost 900 female virgin flies. And we are now sorting these animals by sleep amount. So on the left, you have the ones that sleep the longest. And on the right, you have the ones that sleep the shortest amount of time. Each vertical line is a different animal. And so as you can see, there is obviously a distribution with, between the extremes. And the extreme is where things get very interesting, especially for females. If you look at the female flies, you will see that there's a number of females actually that has hardly any sleep. And these are wild-type laboratory animals. These are not, mutants. In fact, the flies are in principle all siblings among each other. So there is some kind of natural variability that emerges when you analyze a population that is large enough. And that's puzzling, because then we find the same kind of variability that I talked to you about before in those birds, or in general across species, even within animals which to a certain extent share the same genotype. And so the question is, what is special about these animals that sleep so little? So these animals that can cope with only a few minutes of sleep a day, like in these cases here, we have, I think the animals we had where we found the shortest sleep amount were in the range of 20 minutes a day, 20, 30 minutes a day, which is, again, similar to what is been observed in the most extreme short sleepers, like cavefish. So can we, does it mean that we can remove most sleep from any fly, or is just this a prerogative of some lucky or unlucky, if you want to put it, animals that we uncover through our extensive analysis? So the way to address this question is obviously to study sleep deprivation. Study sleep deprivation means depriving the animal of sleep, removing artificially sleep from the animal, and see whether they can cope with it. And that brings us in the second aspect of sleep studies, which is homeostasis. So studying sleep deprivation is not easy. You can, people have been doing this in mammals, in dogs at the beginning of the 1900, in rodents for most of the 1900, and using also systems like this one, which are quite problematic. Here, what you see are rats that have to walk on a treadmill. And the reason why they are so eager to work on a treadmill is that because if they get to the bottom end of the treadmill, they get a little bit of an electric shock. So their paws will get exposed to electric current and they get a shock. So what it means that if they fall asleep, they will get punished with an electric shock. So now this system is relatively efficient, but as you can imagine, it's very stressful. And that's a problem, because sleep is already inherently stressful. Sleep deprivation is already inherently stressful by itself. So if on top of it you also add stress that is due to the sleep deprivation procedure, you're really complicating your life and the animal's life. In Drosophila, most people will do sleep deprivation using shakers. So you put the animals on a laboratory shakers, and then every minute or so you move the shaker so that all the animals experience this kind of earthquake with regular frequency, and that will be enough to wake them up if they are asleep. Again, the problem with the system is, first of all, you are interfering with all animals at once, irrespective of whether they're asleep or not. So you are introducing a stimulus, and a stressful stimulus, if anything, even when you shouldn't. And the other is that mechanically is actually quite invasive. So flies can actually get quite damaged by something like this. So what we did is we took inspiration by a system that was developed in the 60s by Rechtschaffen, and is known as the Discover Water system. So with this system, the laboratory of Rechtschaffen managed to chronically sleep-deprived rodents, rats specifically, not necessarily removing the stress component, but at least trying to control for it. So the way the system works is is depicted here in the figure. There are two rats. One is the experimental animal and the other is the control animal. They are both hooked to an EEG machine, so the computer can record the sleep state at any given second. And when the experimental animal falls asleep, then the machine will command the rotation of the disk on which they sit, which, as you can see, is the same disk for both animals. So the animals will be pushed against the wall of the chamber. If they are asleep, they will be forced to wake up, or else they will fall into water. And rats obviously hate water, so they have a great incentive to just wake up and walk against the rotation of the disk. What it means is that the experimental animal will be fully sleep deprived, or else they will fall into water, which is something that they do several hundred times a day by the end of the experiment. And the control animal, on the other hand, can sleep whenever the experimental animal is awake. So at least for the first days of the experiment, the control animal does get a decent amount of sleep. what happens when you do this experiment? What happens is that experimental animal dies quite consistently. After 21 days of experiment, the experimental animal will just die. And we still, after almost 50 years from these experiments, we still don't know why they die. Staffen thought that maybe it was due to problems with immune response because these animals were developing infections, but even giving antibiotics will not rescue the animals. They will still die after 21 days. The animals show a metabolic systemic syndrome, so their metabolism is completely affected. So they obviously appear very stressed, they lose fur, they eat much more, but they lose weight, and their body temperature changes. Interestingly, the change in body temperature, which happens around day 17, is the point of not return. If you stop the experiment after that point, when the body temperature changes, and you put the animals in the cages and give them all the sleep they want, they will still eventually die after 21 days. So the good thing about this setup is that it reduces the amount of stress that the animals are exposed to by limiting any stimulus to when the animal is actually asleep and not giving stimulus all the time. And in a certain way, you can also give the same stimulus to companion mock animals to control for it. And so what we did is we create robots that can be connected to the otoscope, driven by the Arduino, as I was saying before. And these robots can be programmed to give all kinds of stimuli. For instance, we have robots that can give odor stimuli, so they can send a puff of air to the animals whenever we want to, for instance, when the animal falls asleep. Or they can be used to rotate the tube at different speed. Or they can even be used to rotate the tube and at the same time dispense optogenetic light stimuli to interact, for instance, with optogenetic manipulation. So we use this tube rotation as a way to keep the animals awake, and we end up with an experiment like this. So you can have a look here. We program our robot, and we ask the robot to turn the tube, like in this case for this fly, I could be here in the first one. So I can maybe show you again. Oops. Yeah, so have a look at the first fly on the left. This fly is awake, she's grooming. We told the system, turn the tube after several seconds of no walking, and you will see now the tube is rotating and this fly is responding. So even though she wasn't asleep, in this case, we can program for any kind of activity. We can program, for instance, and say, do rotate the tube when flies interact, or do rotate the tube when fly eat, or like we most commonly do, is do rotate the tube when flies fall asleep. And so we now can then compare conditions and ask what happens to sleep homeostasis, control of sleep recovery, when we deprive animals of different amount of sleep. And so we did exactly this. These are different samples for male flies on top or female flies at the bottom. The different shades of blue or red highlight different conditions of sleep deprivation, meaning that we allow flies to sleep longer or shorter intervals before we actually rotate the tube. So we can tell the computer, rotate the tube after 20 seconds of inactivity, or we could tell the computer, rotate the tubes after 60 seconds of inactivity, or after 120, 5 minutes, 10 minutes, and so on. That way, we can allow flies to sleep for X amount of seconds or minutes, and then interfere with only a specific amount of sleep bouts, for instance. So we did that. And what you can see here is that some flies, which are the ones for which we almost basically activated the tube almost immediately after 20 seconds, they hardly get any sleep during the night. Other flies, these are the controls, will get obviously more sleep depending on the interval we choose. And then we end the sleep deprivation at the end of the night, and we start and look at what happens at the rebound sleep on the following day. So how do these flies recover the sleep they have lost on the day after? And what you see here is that we then quantify sleep rebound. So this is the sleep they try to recover on the morning after. And what you see is that the sleep rebound changes as a function of the sleep interference. So this is the interval that we chose to rotate our tube. Rotate the tube after 20 seconds of immobility, 60 seconds of immobility, under 20 seconds, and so on, all the way to 1,000 seconds of immobility. And what you can see is that the sleep rebound is higher where the interval is shorter, which makes sense because obviously with a shorter interval, flies will lose more sleep. But what's interesting and unexpected is that in all cases, we do see a sleep rebound, which is statistically different from control. So in all cases, even in the cases where we disturb flies after 1,000 seconds of inactivity, we do see a sleep rebound. And that is unexpected. Why? Because flies do not have very often episodes of sleep which are as long as 1,000 seconds. In fact, if you go and look at the data, you will see that in this extreme condition, you're actually rotating the tube only 3 or 4 times per night, which is the average number of long episodes of sleep that flies normally will get. And so what does it mean? It means that even just disturbing the flies 2 or 3 times per night, 3 or 4 times per night, will lead to a sleep loss, which then will require a sleep rebound, a recovered sleep. This is something that is counterintuitive, but is something that we see also in humans, for instance. Sleep disruption can have an effect on sleep quality, which is as impactful as complete sleep removal. Think of a baby or a toddler that will wake you up in the middle of the night just because they want a cuddle or because they want some milk. If you're lucky, this event might last only a few minutes during the night, and yet it will be enough to make you feel rather miserable in the morning after. So the way you feel on the morning after is not necessarily linear to the amount of sleep you have lost. And we see this to an extreme in this case. So this is an experiment that was done in the 60s or 70s, if I remember correctly, the late 60s, and it's known as the Randy Gardner experiment. It is an experiment that was done by Randy Gardner, who was a American high school student in California, and he wanted to participate to a science project, a science fair, and at the same time, he wanted to enter in the Guinness Book of Records. And so what Randy did is he stayed awake for 11 days in a row, under attentive control of his friends and medical doctors. There is a BBC podcast that you might want to listen to, which I strongly recommend. It's called The Boy Who Stayed Awake for 11 Days, which retells the story of Randy as seen by the people who were there to witness what happened. And so Gardner stayed awake for 11 days without ever sleeping and without using any chemicals to stay awake, so no coffee allowed. During these 11 days, he will be tested in several fashions to check for his performance. He will play basketball, he will play chess, and he will do pretty well. Every now and then, he would experience bad episodes of nervousness, aggression, and even some hallucinations. But overall, he did okay. And then after 11 days, it was decided that the experiment was to stop because the goal of entering in the Guinness Book of Records was achieved. And so Gardner then went to bed at night after 11 days of sleep deprivation and woke up the day after, recovering only 2 or 3 hours of sleep more than he will normally do. So after 11 days of continuous wakefulness, he did not have to recover, the equivalent of 6 days of sleep. I did not stay asleep all week following. You stay asleep during the recovery period only for a few hours, and then he was as fresh as new. He actually went to school the day after. So that's something, as I said, we see only in humans. Same thing we saw, similar thing to what we saw in flies. That the recovery sleep that we get is not necessarily a linear function of the sleep we've lost. And if you think about it, it does fit, again, the tripartite model of sleep function, because it tells you that the sleep you have to recover is only the one that is vital and possibly the one that is useful, but definitely not the one that is accessory. So what happens with flies? We've done the same experiment. We did the Randy Gardner experiment with Drosophila. We started chronic sleep deprivation when they were about 7-8 days old. And then we went on for, again, about 10 days, same as Randy Gardner. And we stopped the sleep deprivation after 10 days. And then we look at rebound on the morning after. And if you look at the rebound here, comparing the male, in this case, or the females to their control counterpart, you will see that there is clearly a rebound. So the animals that have been sleep deprived, they sleep longer the morning after. The females also sleep a bit longer, and interesting enough, for females, the rebound continues and can be observed also during the following night and the following days. While in males, it actually disappears. They go back to normal already from the first night, basically. And so the rebound is nothing to write home about. They, like Randy Gardner, also flies. They are okay, recovering just basically a couple of hours of sleep after such a long sleep deprivation. So these matches, as I said, and can be explained conveniently by the tripartite model, by the fact that flies do not need to recover any of the accessory sleep, which account probably for most of the sleep. They might have to recover some of the useful sleep, but not necessarily. And probably the only sleep that they really have to recover is the elemental one. But also keep in mind that in our case, we do have short periods of sleep throughout the length of the experiment, because we allow flies to rest for 20 seconds every time. And so one could argue that those 20 seconds of sleep that they get now and then is probably enough to account for at least some of the elemental aspects of sleep. And if that's the case, if that's the hypothesis, then the question is, will flies die when they are sleep deprived for very long, like it happens to Rechtschaffen rats, or will they be fine even with chronic sleep deprivation? And so we did a long experiment of chronic sleep deprivation, where we, again, started the sleep deprivation when flies were relatively young, a few days old, and we went on for as long as we could, basically as long as the flies were surviving. And when we analyze longevity in these animals, what we see is that there is no statistical difference in male flies. So male flies sleep deprived for the length of their life. They sleep as long as the control counterparts. And there is very little difference. There is a little difference, statistically significant, but it's not much, in the range of few days for female flies. So that tells us that most sleep does not really have a vital function. And there is only a little component of sleep that is vital, and you can see it probably here in the flies, in the female flies dying. Or maybe the reason you don't say with male flies is because they're better at coping with those 20 seconds of inactivity and concentrate the vital function of sleep at those times. All right, so we've covered the sleep amount, we covered sleep homeostasis, and we have uncovered some interesting general principle of sleep function, which I believe can be applied to all animals, not just to flies. And again, on this line, let's finally cover the last aspect, which is sleep quality. So as I say, sleep quality is often made to coincide with sleep reversibility, at least in the way we measure it. And when you think about sleep reversibility, and so the ability that we have to respond to external stimuli and react to them by waking up, obviously, then you start thinking about the fact the brain is not fully off during sleep, right? Which is something that we know very well in the field. The brain is not dead. You're not brain dead during sleep. You can always wake up. And you will wake up with stimuli which are, dependent on the conditions in which you are. You could wake up when challenged with a very loud bang, for instance. So stimuli, they have a strong quantitative component. So you might not wake up if your cat moves or walks in the room because the noise is not loud enough. But you might wake up if there is a car alarm going off in the middle of the road, because that triggers a certain quantity threshold. But at the same time, it's more interestingly, you will wake up in response to qualitative stimuli too. So what is a qualitative stimulus? A qualitative stimulus is something like your name. So it's been shown in the 60s in this seminal paper that if I call your name while you are asleep, you'll wake up. If I call somebody else's name while you're asleep, you won't. So if I record your name and I play back your name during sleep, you'll wake up. But if I play back the same tape backwards so that the same stimulus makes no sense, you will not wake up. And this is what they've done in this experiment. You see here, Sally will wake up, a person called Sally, but the same name spelled back will not. And in this case, I think Maya will wake up Maya, but all the other names will not. We know that this is true in animals as well. This is an example of anecdotal evidence that you can find on YouTube or TikTok, like I did. Animals waking up to olfactory stimuli that are being prompted by their owners during sleep. There's plenty of these examples, not really very well investigated in the literature, but this is one of the things why the internet is interesting. You can find these evidences on YouTube and TikTok and Twitter, and they are common sense. So we decided to do something similar with flies. We wanted to see whether others can also wake flies up. Obviously, we cannot really call them by their names, but we can use others, like you've seen in other animals like cats and mice and porcupines and so on. And so we use this robot, the first one on the left, which is the robot that is able to trigger a stimulus of olfactory nature. So we can basically give odor, purple odor or a blue odor, and then by changing the valve, we can decide which of the two odors will be delivered to the animal. So this is then connected to the otoscope. The way it works is that the animal is in the tube. Whenever the animal falls asleep for a certain amount of time, this will trigger back to the Raspberry Pi. The Raspberry Pi will activate the robot, and the robot will give a puff of air or a puff of whatever substance we want to test. We've started testing this with acetic acid. The reason why we went for acetic acid is because it's well known that acetic acid will elicit different responses in awake flies depending on the concentration. And this is an example of flies that have been put in something we call an olfactometer. So each dot here is a fly in a position. In this, you can about make up the shape of the space in which they're placed. If we give them low concentration of acetic acid, so equivalent to about 1% of acetic acid in water, they love it and they all try to go towards it. But if you give them high concentration of acetic acid, they are rather neutral to it. And the reason is believed to be associated to the fact that acetic acid is a natural compound of fruit, which also gives information about the ripening of the fruit. Fruits that are more appealing to the flies will have lower concentration of acetic acid. As the fruit matures too much and will become rotten, it will lose that attractiveness. So it's interesting because it's one of the stimuli that qualitatively will be more attractive at lower concentration. So if you compare this to a bang, this is a stimulus that where the softer bang is more attractive than the louder bang. So we give animals that for different concentration of acetic acid, either attractive in blue, so 1% or 5%, or neutral in red, so 10% or 30%. And then we measure their probability of arousal per puff. And as you can see, there are times during the night where there is an increase in arousal with acetic acid compared to air. And if you go and quantify this at different time points, the picture becomes clearer. For instance, during light night sleep, so light sleep, you'll see that acetic acid at 1 or 5%, so the positive concentrations, will wake up flies, but acetic acid at 5 or 10% will not. And during deep sleep during the night, actually all of them kind of reduce the ability to wake up. And also during the siesta, you see again that the ability to wake up is associated only to the positive concentration of acetic acid. So what does it tell us? It tells us that animals like flies can detect a qualitative information in an odor they perceive during sleep. So there is some level of their brain which is able to qualitatively distinguish the information of a sensory input during sleep. And how is this modulated? We looked into that. We started interfering with animals and then study how different odors will be behaving differently in different conditions. One thing we did immediately is exposing flies to ethanol. So you can give them a beer and get them drunk, or you can just give them water and then probe how they respond after this event, and you will see that they lose. After the beer, or well, after ethanol, in fact, they lose partly the activity to wake up to acetic acid. You can sleep deprive them. So you can either test animals which have been rested throughout the 12 hours of the night, or which have undergone sleep disruption during the 12 hours of the night. And then again, what you see is that there is a difference in waking response in animals that have been sleep deprived. And you can also selectively test odors in a condition of food deprivation, for instance. So what we did here is we starved animals for several hours, and then we probed arousal during the night using odors which either had or had not a food valence. And so here you see odors which are not related to food, or odorants which are related to food. And you see that most of the odors which are related to food confer in starved animal an increased response. So starved animal are more likely, as you see also in panel C, to recognize and wake up to odors if they are related to food. Starved animals will not recognize and react to odors which are not related to food. So again, that tells you that there is a specific qualitative engine in the brain that in this case is reasonably triggered by food-related odors, and reasonably because the flies have been starved preemptively. So we can start putting together the piece of the puzzles and start looking at the ways by which the sleep and subconscious processing information can be modulated by odors. We also looked at the slide is missing, but we also looked at the circuit that highlights this. And we explore what are the brain connections that are connecting the fly nose all the way to the sleep centers of the brain. Okay, so finally, in the last 5 minutes, because I'm really running late, I want to go into some unpublished data which look at all this problem again from the evolutionary point of view. So we kind of close the circle, go back to evolution. And what we did is we looked at sleep in different Drosophila species. So we picked 7 in total Drosophilids, so not just Melanogaster, but also Simulans, Eschelia, Erecta, Jacuba, Winlid-Stony, which are all fruit-feeding Drosophila, and Virilis, which is an example of vegetable-feeding Drosophila. They span different areas throughout the world. And we looked at a sleep pattern on the left here, or activity pattern on the right for all of these species. We also looked at wild-caught Melanogaster for good measure. And what you'll see here, without going into much detail, is that more or less in terms of quality of sleep, all the Drosophila species have the same pattern. So they all sleep mostly at night. They all mostly active at sunset and sunrise. And in all cases, the males have a kind of a siesta during the afternoon. So that is interesting. It tells us that sleep is conserved between Drosophilids from this point of view. That's not unexpected. One could argue on whether sleep amounts changes depending on whether the flies arrive from certain latitude or other latitude. But instead, what we did is we then looked at homeostasis of sleep. So we sleep deprive these animals using the tube rotation paradigm. And you sleep deprive them throughout the night as we did before. Obviously compare them to the same species but in control condition. And then, as we did before, you look at sleep rebound at the beginning of the day after. And you see here In the top panel, the sleep rebound in Drosophila melanogaster, either the lab strain or the wild caught, is highly significant. The day after, the first 3 hours of the, or 6 hours of the day after, you get an increase in sleep. So you get a proper functioning textbook sleep homeostasis after sleep deprivation. But interestingly, that was seen and observed only in Drosophila melanogaster. We could not see anything like this in the other species, none of the other species. So that was very unexpected, very puzzling, worrisome to a certain extent. So what does it mean? Does it mean that sleep is not a conserved feature of all Drosophila? So to look into that, we then use a different system of sleep deprivation, which is sleep deprivation by social stimulation, an experiment in which you put 2 male flies together, they fight, they interact, they keep each other awake during the night. And we've shown this, that this gives a strong sleep phenotype and rebound in melanogaster. And interestingly, when we do this different type of sleep deprivation, we see a rebound appearing at least with some of the species. So with Yacuba, with Seychellia, with Simulans, we do see rebound after this type of sleep deprivation. So we still do not see rebound in Erecta, in Willistonia and Virilis. So not all species show rebound after social sleep deprivation, but some do. So that tells you that probably what's happening is that homeostasis is a component that is common to most species, but is triggered by different natural or ecological stimuli. One thing is to stay awake, and rebound because you've been partying all night, and another is because you've been fighting all night or because you've made love all night. So all these different ecological stimuli might trigger different components of the rebound machinery. And finally, and this is my last slide, we also looked at the conservation from the pharmacological point of view. We give animals drugs like L-DOPA, which is very well known in the literature to interfere with sleep regulation, or chlordiaform, which does the same. Look what happens when you give L-DOPA to Drosophila melanogaster. So the control flies will sleep, as normal, about 50% of the time. And then you give them, this is during the night, and then you give them L-DOPA, and boom, there's hardly any sleep. So this is well shown. There's plenty of papers that show a role for dopamine in sleep regulation in Drosophila. Interestingly, the same treatment did not elicit anything similar in any of the other species. And that was true both for L-DOPA and chlordiaform. So mostly for chlordiaform, exception for Virilis. So it tells us that really in these animals, it is sleep appearance, sleep pattern, the sleep amount, if you wish, that is conserved. But the underpinning and the underlyings that regulate homeostasis, or in this case, the neuropharmacology of sleep, possibly have evolved independently. And so we might be seeing cases of convergent evolution of sleep if we look close enough and we go beyond the classical, analysis of sleep amount or the classical analysis of, EEG or whatever. If you do look at homeostasis, at the neuropharmacology, at the genetics perhaps, then you start seeing the traits of sleep that have evolved convergently. And again, this goes in favor of the tripartite model, because if you think about the tripartite model, then you can think how, the basic drive of sleep, which is the one really driven almost totally by the accessory component, is conserved apparently throughout evolution. But then other aspects, which are secondary, at least from the analysis point of view, might not. So that I close it here, and I want to acknowledge the people who did the work. Most of this stuff about the sleep amount and sleep homeostasis is been done by Quentin, who is now assistant professor in Denmark, and Esteban, who is now as his own lab assistant professor in Buenos Aires. And the work on arousability and others been done by Alice, who is currently at the Creek. And these are some other of the people who've contributed and who are still in the lab. So thank you. That will probably run a little bit long, but happy to take any question if there are now.
Great, thank you so much, Georgia, for a really interesting and truly amazing talk. And it's also great to learn that Quentin, Esteban, and Alice are doing currently great things as well. So I'll just take the privilege of asking you the first question, which is in the graph that you mentioned before about lifespan in flies, sorry, about sleep in flies across lifespan. Are there any other features of
This
there
one.
that no, the other one actually. So is there another relationship between, for example, sleep bout, the number of sleep bouts, the number of sleep duration?
No, we never really saw any correlation, any strong correlation between sleep amount and longevity. So we never really saw that flies that naturally sleep less will live less, or vice versa. So in general, we also have mutants like the fumin mutant, which is a mutant where sleep is reduced almost completely. These flies have only 1 or 2% of the normal sleep they normally get, and they just live as long as normal. So it's always been very difficult to find any correlate between sleep length and longevity.
Cool. Thank you so much. And I was wondering what your thoughts were about the sensory processing during sleep. Is there like a biological underpinning for that, like a biological circuit?
Yes, yeah. Unfortunately, I don't know why the slide was cut off, but we do provide in the paper, which was published on Nature a few months ago. We do provide, in a way, the first probably diagram blueprint of a circuit that explains exactly this. So it's a circuit that connects the olfactory receptor neurons all the way to the dorsal fascial body, which is an area of the fly brain that we think is responsible for sleep control. So obviously more can be studied. In a way, Drosophila is an excellent system to study this circuit. I don't think it's by chance that we basically show one for the complete one for the first time. It is because neuroscience in Drosophila can really be done to an extent that is very difficult to to compare in other animal models.
Cool. Thank you so much. And I was wondering if you had any advice for students and postdocs and how to go on and to achieve great things in terms of the sciences.
In terms of science in general.
Yeah.
Oh, well, that's might take another hour. No, well, yes. I would say one thing that I can been discussing recently about, it might be interesting to students watching this or people who want to start a PhD, is to focus on the environment more than the project. So that's something that I think is not stressed frequently enough. So if you want to start working in science and want to start a PhD or something like that, do not get too constrained about the scientific project you're going to follow, the specific question you want to follow. Chances are that in 5 years, you will be working on something completely different anyway. And so do focus on the environment, a good lab, a good department, a good institute, good colleagues. These, I would say, is the most important things in terms of picking a career in science.
Cool. Okay, wonderful. I'm not seeing any more questions. So on that I think someone raised their hand. Dr. Giorgio Fedele. I'll I guess allow you to talk. Does that work? If you want, you can unmute yourself. If that's okay.
Hello. Yeah. Can you hear me?
Yeah.
Yeah. Hi, Giorgio. Very nice talk.
Thank you.
Just a quick question. Sorry, I was driving, so I couldn't really see the slides. Anyway, yes, yeah. You know the final experiments that you did with the different Drosophila species.
Yes.
Were they wild caught or were they coming from stock centers? Because I was wondering whether you try to, because of course they are coming from different latitude, so they will be exposed to different temperature cycles and different photoperiod. And I was wondering whether you guys have tried to change the photoperiods according to the latitudinal origin of those species and see if the sleep changes in any way, or if you can see any differences between those species and Melanogaster.
So the species were wild caught, and we had two different lines per species. So two different, differently wild caught animals lines per species at different places, different time, except for erecta. I think for erecta we had only one. So that's answered the first question. The second question is no, we didn't do anything with the photoperiod. I know that Charlotte is working on that, and that's not something that we are, it was particularly relevant for our specific question. So no, we didn't do anything with the photoperiod.
Okay, well, thank you very much.
Well, thank you. And on that note, I think I'll take the privilege to thank you, Giorgio, again for a really, truly wonderful talk and for some great advice. And to all of the people listening on YouTube, thank you very much for sticking with us, and hope that you'll come to our next talk. And for that, please do have a look on our website, internationalsleepcharity.org. And thank you again, everyone, for attending, and thank you, Giorgio, for the talk.
Thank you, Yusuf.
Thank you.
Thank you.
Bye-bye.